The next project in our competition is titled A Comparison of Toothpaste at Bacterial Inhibition. The aim of this investigation was to find out which toothpaste would be better at inhibiting bacterial growth. Arvids ANDERSONS will now take the floor.
Hello, my name is Arvids ANDERSONS, and today, as you've just heard, I'll be presenting to you my investigation. My investigation involved looking at different toothpastes aimed at different demographics to address the issue of childhood caries. I looked at the toothpaste and saw how they compared when inhibiting the growth of Lactobacillus casei, a bacteria. Behind me are a plethora of flags. Now, these flags represent my global and international background. But you know what else has an international and global presence, or even background? Caries. In fact, 52% of children, ages 6 to 11, have caries in their primary teeth in the United States of America. The World Health Organization deems dental caries the most common uncommunicable disease worldwide. But why is this of interest to me? Why am I interested in this issue? And why did I decide to explore it? The answer is really simple. I have an inherent interest in dentistry as a prospective career. And of course, in dental school and beyond, I'll be expected to perform my own research, whether it be passion or business. As such, I saw this opportunity to conduct my own research, my own investigation as, well, the perfect opportunity, as I'd be able to not only develop my skills in the lab at an early age, but also get to read scientific journals, analyze them, and increase my knowledge in biology in general. So before proceeding with the method, the results, etc., I'd like to introduce to you the toothpastes that were used. So here's a data table, an extract from my paper. The first two toothpastes are both aimed at the 6+ demographic. The first toothpaste is aimed at the 6+ demographic and contains a lot of green, natural ingredients. The second toothpaste is also aimed at the 6+ demographic, but contains industrial, more traditional ingredients. I thought it would be pretty interesting to compare these two toothpaste aimed at children, one with industrial and one with natural ingredients. The final toothpaste is aimed at the 12+ demographic, so adults in the dental world. I decided to compare it to see if there is a difference in inhibitory qualities from 6+ toothpaste and adult toothpastes. One thing that should be noted is an outstanding material in the third toothpaste, so a differential, is stannous fluoride. This will be an interesting talking point later. So the method can really be broken down into 3 major steps. So step 1 was creating the liquid growth medium, or the broth. So to do this, I followed the parameters on the third box to create the broth in which the bacteria would grow. Once I've done this, I used an inoculation loop to scoop some solid pre-prepared stock of Lactobacillus casei. I then mixed this in a flask and put the flask in an incubator at 35 degrees Celsius to let the bacteria grow. Once I observed no contamination and some growth, which I checked with my supervisor, I knew I was ready to create the agar and the petri dishes. To create the agar, again, I followed the parameters on the bottles. And once the agar was ready, I poured it into the 8 petri dishes. I let it solidify, and then I used a cork borer to create 3 holes. Before creating the 3 holes, of course, I drew a vertical line and a horizontal line on the back of each petri dish, splitting it into 4 quadrants. So 3 of these quadrants would be occupied by each of the toothpastes. The 4th quadrant was a control. There would be no toothpaste. I then put all the petri dishes in an incubator at 35 degrees Celsius. The final step, of course, was to gather the results. Data collection. So I did this by taking a picture of the Petri dishes for each day. So here you see day 1. This is the first day of the pictures, and day 5, so the last day of the pictures. Now, you will notice these red dots. This is not the natural color of the colonies. That would be, I guess, pretty concerning. No, these are markings I made with my Surface pen to manually count the bacterial colonies. I did this because there was no clear zone of inhibition, a traditional method in these type of labs. As you can see, this is my raw data. It's pretty intimidating. So the reason for, if you look in the third column, I have M's and S's. And this is because the bacteria were not homologous in their nature. Some of the colonies were big, some of them were small. As such, when I counted them, I made sure to count the amount of medium, big, and small colonies and awarded respective points for each. Medium colonies were worth 4 points, big colonies were worth 16, and small ones were worth 1. I then added this up to find the colony growth score. I then graphed the colony growth score for each of the quadrants after 170 hours of growth, which is the total amount of time. As you can see, some of the data is pretty close. For example, the two toothpaste aimed at the 6+ demographic, PS, Schmelz, and G, green, the one with the natural ingredients, were pretty close. One minute. When you factor in the error bars, you would see you needed to perform some statistical test. So I performed a Mann-Whitney test for all trials against the control and the 6+ toothpaste. I found that all failed the test, not showing statistical significance, except the control against Oral-B. This made me curious to investigate the other toothpaste against Oral-B to determine if there's really statistical significance. And I found that yes, there is. So the conclusion that can be drawn from this, the only firm conclusion that can be drawn from this, is that the toothpaste aimed at the 12+ demographic, Oral-B, was more successful at inhibiting the growth of Lactobacillus casei than the other two toothpaste aimed at the 6+ demographic. I believe this is a rather good conclusion. As earlier I mentioned, the outstanding material was stannous fluoride. In my dental school and in my future research, I think this can be an incentive or a basis for future exploration to see how stannous fluoride can either be implemented into toothpaste aimed at the 6+ demographic, but weighing the benefits and the risks, or seeing if stannous fluoride was really responsible for this increase in inhibitory quality. Thank you very much.
Please welcome back.
Hello, and thank you very much for the presentation earlier. We have some questions to ask. So basically, it would be interesting to hear what was the starting point for your studies on toothpaste?
Yeah, so I mentioned in the presentation that I'm interested in dentistry as a prospective career. Next year I'm going into dental school, and then afterwards, of course, I'll be working as a dentist. So I thought, well, I should already pursue this passion. I should look at things I can already look at, toothpaste and etc., because I use it every day. So I was interested to see the chemicals inside of it and how, of course, it works to inhibit bacteria.
Yeah, sure.
Is there a specific type of toothpaste that you found in your research that you would recommend?
Yeah, so what I tell people when they ask me this is just any toothpaste with stannous fluoride, to me, is good, because I did another lab where I was investigating toothpaste, putting it in different sodas and just seeing how the enamel erodes. And through that, I was doing a lot of research online, and I found that one common denominator is that stannous fluoride is really good at protecting the enamel. And of course, even when you have bacteria, their byproduct is what causes the issue, acid, and stannous fluoride will protect it. So when you look at toothpaste, look at the back and look for stannous fluoride. It's similar in all languages, stannous fluoride. So that's my recommendation.
Very good advice.